So hello everyone, thank you for joining. Today, Rick van der Zedde and Pieter de Visser will present the seminar. Rick van der Zedde is they are both from Wageningen University, working at the plant science group. And Rick van der Zedde is working on the automation and robotics part, phenomics part. He's specialized in computer vision and robotics. And Pieter van de Visser is working on crop physiology. So both in Wageningen. And they will present this very, I think, interesting way of making plant modeling and artificial intelligence and robotics and computer vision discussed together. So the floor is yours, and thanks again for accepting to participate.
Yeah, thank you. So Pieter, you can do the first part. I'm ready.
Yes. Thank you, David, for our introduction and to invite us here. I will first, Pieter de Visser, I will first do the first part on plant modeling and digital twin virtual plants, and then Rick will follow up with more stuff on artificial intelligence, robotics, etc. It's about horticulture, where we both work in, about digital twins, virtual plants, and AI. You can see on the first slide already that you have some virtual digital representations of plants. That's where it's all about. Next slide, please. I'm having a background in 3D modeling of plants and plant physiology. And I'm working on a greenhouse horticulture business unit. And Rick, well.
Automated slide transition, my fault.
Yeah. We saw that Rick has a specialism in artificial intelligence and nowadays working in phenomics and automation. Okay, so we are both from Wageningen University and Research, and you can see that on the next slide. We have a nice campus, and we are quite an international organization. We have students from very many countries, and well, we have a university part and an R&D part. We are from the R&D part. Okay, next slide. That is about the project Rick and I share, the digital twin on a tomato crop. So you can see here, you have the twins. You have at the left in the graph the photo of a real crop growing, putting sensors inside of the greenhouse, getting information from those sensors, getting those data towards a computer model, virtual tomato crop, where we have a greenhouse climate model and a plant model together. We do simulations with the input from the real twin, and we give back information on the growth status, and we give recommendations and decisions. And the plan is in the future to integrate that in a decision support system where growers and breeders can profit from and can possibly use that in their greenhouses. But for now, it is an experiment, and we're going to show you a couple of things of that experiment. Next slide. The basis of the 3D plant modeling, that is my specialism, is L systems. Maybe you have heard about it. L systems are already invented in the late 60s by Professor Lindenmayer, and it is a sort of modeling where you have all the organs as branches. You have a sort of branching structure. You simulate. You can have infinite number of plants in your scene. You can, apart from modeling the branching and so on, simulate climate like ray tracing, spectrum, temperature, etc. And when Rick clicks on the graph, you will see gradually a simple plant appearing. So it gets branches, and then, yeah, basically that is the basis. But you can have very fancy plants in the end. You can see here the rows, or sort of rows. And the next slide, you can see there are quite a lot of plant species that have been simulated already with L systems. There are various packages, software packages you can use for that. But you can see here examples on the tomato plant, the chrysanthemum. Trees are often modeled also with this approach. Cucumber in the middle, and even tissues of organs where it's the cell as a basic unit can be modeled. So you can do quite a lot of things. It is very versatile. Next slide, you will see that we can model a tomato crop in a greenhouse. I don't know if you ever visited a tomato greenhouse, but the plants get very tall. And there is now in Holland a lot of things to do about LED lighting within the crop or on the top of the crop. And you can easily model in 3D the structure of the plant, the rays casted by the LEDs. And you can see to the right two types of plants, two genotypes with different leaf patterns. And that, of course, affects the light interception and the growth of the plant. In the left, you can see an inlet where you have a small, tiny plant with the colors that indicate the light level that falls on the specific places in the plant. So with this software, we can very accurately estimate where the light comes and its consequences for photosynthesis and for growth. Okay, in the next slide, you will see that we now have a sort of 4D approach. It means you can see the plants grow in the greenhouse, and that is the digital twin about. It is basically what we call a functional structural plant model, FSPM, where we have the function like photosynthesis and growth, and the structure like the geometry of the leaves put together and make it into a living digital thing. And you can see by clicking, yeah, that we have now, as an example, 6 plants growing there, underneath a couple of lamps. And basically, this is what we need for our digital twin. Next slide. You can see here, I indicate that the FSPM model needs a lot of data. So per organ, we want to know its size, its ripeness, its volume, the angles, the length, etc. All those architectural things you want, also the physiological things. And you can imagine that for a whole greenhouse, you want to know of each organ its state. And that is what the digital twin is about in our approach. Okay, in the next slide, you will see that in the end, we have a greenhouse climate model to the left, where you see for 2 days of animation, the temperatures inside the greenhouse, CO2, the air exchange in the roof, things like that. They are all based on a real biophysical model. And to the right, you see the rendering of a digital tomato crop inside of the greenhouse, completely 3D, where we have a multispectral casting of light rays. And these two bigger models are merged together for our digital twin. And the next slide, you will probably see how that works. You cannot see and read all the details, but if you look at the icons, the figures, you can see the 5 most essential things in our complete design. To the left above, you see RayPro, and that is a model that calculates the intensity and the direction of the rays that come into the greenhouse. And those rays are then intercepted by the plant model that is in the middle a little bit. That is called GrowIMP. GrowIMP is the software platform we use for that. So the plants catch the light and process it for photosynthesis. At the same time, to the left, you see the image of Caspro. Caspro is, of course, simulating the climate in the greenhouse, the temperature, the humidity, the CO2, very important input for the growth model. So that together gives a certain production, and that information is passed to the control system, and it is gathered together, put somewhere in the cloud, and Rick will talk about that later. And that information is then supplied to the grower. And in our case, in our experiments, it's still an experimental growing condition. On that basis, we can change the climate in the greenhouse. For instance, open the windows because it is too hot, or put out the lights because the light is sufficient for growth. And then the cycle is around. And then again, we're going to look to the data and the status of the crop inside of the greenhouse. And that is in the middle. You can see the video camera. It's the NPEC camera. And that system is crucial to get all the data we need. And I will now give the word to Rick. Rick, there are not yet any comments, so you can go ahead.
Okay, yeah, so everybody feel free to make a comment or suggest or put a question. So as Pieter explained, we can model a greenhouse and we can model crops and the incoming light. And we decided to put this in a real greenhouse, but then still it's actually a research facility, because we would like to have more control over the plants than a regular greenhouse is able to gather. So I'm gonna first explain this artist impression of our greenhouse, because this is what we call the NPEC Greenhouse. Netherlands Plant Eco-phenotyping Center is funding this greenhouse. And the essence of this greenhouse is that it's completely fit for doing gathering data around the phenotype of a plant. So these two compartments, they are, for instance, the tomato plants growing. And we have around 1,200 plants growing in these two compartments. And they are actually placed on conveyor belts. What this means, we're doing this because we are able to move these plants on long lanes while we are then bringing them to this area of the greenhouse where it's filled with imaging systems. So we are able to make pictures in an automated way, but also in a quite a controlled way. So we have a range of cameras in here. I will mention a few in the upcoming slides. And in this way, we can collect information that can be used to give back to the digital twin. And then the virtual world can be optimized by measurements in the real world and vice versa. I'm going to mainly talk about this part of the greenhouse, because at this part of the greenhouse, we decided to actually go towards another measurement principle. Here, the sensors are moving to the plants. So here the plants are on tables, and we are gathering the phenotype while bringing, for instance, these kinds of scanners to the plants. That's actually another topic not covered today. Short about NPEC. NPEC is actually funded by the Dutch Science Organization, NWO. We have 22 million euro to build this research facility. And the digital twin is one of the projects that is making use of the fact that we have this greenhouse. But any project, you also, including also industry, is able to make use of the fact that we are building such a research infrastructure. NPEC is not only the greenhouse, actually the greenhouse module is number 5, but we have also been setting up field phenotyping tools, so drones and a field explorer, also climate rooms to study the phenotype of a plant in a range of conditions. And actually the lower we go into the module numbers, the more control we have, but also the more precise the measurements are. And also the more controlled the environmental conditions are. In this case, we're talking about ecotrons, in which we have a small population of plants growing in super controlled conditions to study, for instance, plant microbe interactions in soil. And also, we're going to focus on number 5 for now. And you can imagine that all these systems produce enormous amount of data. So as soon as I'm going to talk about data, the numbers will come back, these 6 modules. So number 5 is a greenhouse. So it's actually a greenhouse, relatively small, like 30 by 30 meters. 1,000 square meter of greenhouse, and it's actually ready. So we are at the moment installing the last bits and pieces, especially on the high-tech tools. So the equipment is being installed and tested at the moment. So this is what I've mentioned earlier. These are the conveyors. So each tomato plant grows in a pot, and these carriers are moved on these green conveyor belts. So they are able to be moved, for instance, for automated watering over here. These are the air tubes to give fresh air to the whole compartment. These are the LED lighting systems to give light to the plants when the sun is down, for instance, to give simply additional light later in the evening, afternoon, especially in the wintertime. And all the imaging systems are back there. I'm going to also address them. I have another impression, actually, because these are the big boxes in which all kinds of cameras are hidden. So these are, for instance, chlorophyll fluorescence imaging done over here with a long dark adaptation tunnel where the plant goes in and takes about 20 minutes until they are measured and fully dark adapted. Over here we've got the hyperspectral camera system, and at back a 3D camera system. So I'll mention only a few. I'm going to mention the hyperspectral camera. So that's in one of these big white boxes, which is actually a camera by IMEC company, also making use of Sony chips, actually, and also their own chips. And it's actually funded by the One Planet Research Center, which is an initiative on our campus where we have teamed up with IMEC to bring high-tech closer to the agri-food domain. And if we talk about hyperspectral imaging, then for instance, we are focused on spotting weak elements on leaves that could have a strong correlation with, for instance, early emergence of diseases. Just to give you an example, then if you have hyperspectral data, it's not an image, it's actually a multi-channel image, not 3 channels like RGB, but maybe up to 200 or 300 channels. And we have all kinds of software tools to visualize the data, but also to do AI on the data and figure out which characteristics in the data link to specific aspects, diseases or plant parts, whatever. So talking about the conveyor system, so this is a drawing to give you more insight. So we have 2 compartments that are completely identical, and both are able to give plants to the imaging systems. Just coming back to this drawing, but I think I've explained this in sufficient detail. So the essence is actually capturing the phenotype. I'm not sure about your background, of course, but in our institute, we have, on the one hand, people working on technology. So they are programmers, software developers, and familiar with data handling. But on the other hand, we also have a lot of biologists, plant physiologists, that are less aware of what is doable, measurable with these new fancy tools. So 2 years ago, we bought already a phenotyping system, which we call, and I think everything should have a name, the PSI Robin, which is a pilot device to study this plant to sensor approach. So we can generate 3D point clouds like this one. And in this way, we can also introduce this digital phenotyping to our own community, actually. And this is where we have started. So you have a plant, in this case, tomato plant. You generate a 3D model. And for instance, you are going to measure things in this 3D point cloud. For instance, measuring the height, or measuring total volume, or trying to extract individual leaves from this model. And those are the parameters that are required for the digital twin. I'll show you this one first, but actually in NPEC Greenhouse, I'm going to talk about the MaxiMarvin, which is a second system, also able to generate 3D models. So the PSI Robin is a standalone box on wheels. It's actually quite a small box, about 2 meters high, with a range of cameras, actually. So we got, for instance, a 3D laser triangulation system that generated the point cloud that I've shown on the previous slide. but also like an RGB side view camera to take an RGB picture. As well from the side as actually also from the top. So there's an RGB camera in this panel. What's actually more special about this PSI robot, that it's also a chlorophyll fluorescence imaging panel. So all the light bulbs are meant to give a light treatment to the plant, and then the camera is analyzing what the plant is actually doing with the light. So the light is not meant to make a beautiful picture, but it's really to give the plant a treatment and analyze the response of this plant to the treatment. But another technique that we are using in NPEC Greenhouse is actually what we call the Maxi-Marvin. And in this case, it's not a laser triangulation approach, but it's actually a multi-view approach. So we have 15 RGB cameras mounted, as you can see, a few of them on several levels. Plants go in, doors close, 15 images are taken simultaneously, and then a 3D model is generated. So I've added 2 slides to explain the mechanism. So it's based on volumetric intersection, and you'll end up with 3D voxel cubes. Just to explain this to you, if you're not familiar with it, because I think it's actually quite a powerful and super fast 3D technique. It is worth exploring also if you are in a hurry, actually, because within seconds you can generate quite an accurate 3D model. So actually, you can say each camera is focused on a measurement spot in this cabinet, and all cameras take a picture at once, at exactly the same moment in time, so the plant does not even need to stand still. So you could do it on a moving plant conveyor approach. So just to show you how fast it is, we have been implementing this for literally a decade. And we are generating with this multi-camera approach a picture, so a 3D model, which looks like this model on top. And as soon as the picture is made, as soon as the plant is plugged up, then a robot comes in and takes a leaf sample from one of the leaves that is best approachable. This is an application specifically for breeding companies, where one of the very dull tasks is to take a bit of leaf from the plant, put it into a microplate. This microplate is then brought to a sequencer to extract the DNA profile, while the plant actually continues growing, and then they can link from the genetics certain behavior of the plant. So the phenotype is linked actually to the genotype. So I'll show you the robot. So now a 3D model is generated. The approach angle is calculated for the robot. So the robot knows exactly how to reach out to this leaf, take this tiny leaf sample, and puts it down there in the microplate. So this is a commercial application sold by a robot manufacturer, you can say a system integrator. And we have designed the software only in the system. So we are not selling this robot. We have given this 3D Marvin approach to this machine builder in a license agreement. And I thought, because you are the computer science lab of Sony, I'm going to show you a bit more. So the 3D model that this robot is using is actually a voxel cube representation. So this is one of these models of a tiny tomato seedling. So this seedling is only 8 centimeters high, 8 centimeters in diameter. This is the raw data that is coming from this volumetric intersection approach. So we don't have any information yet. And then the challenge is to figure out what does each voxel represent. So is this particular tiny cube part of a leaf or part of a stem, main stem? So this is actually the outcome of the algorithm. So we are constantly trying to optimize algorithms to figure out what are the plant parts. In this case, the color indicates the type of plant part. So stem, leaf 1, leaf 2, leaf 3. And this is also where the AI actually comes in, because that's, you can imagine that this is something that you could train. So we have published last year a paper on this approach, where we say we have a multi-view camera. So now you understand what this is about. This is the Marvin approach. We have multiple views. We can generate, for instance, a 2D picture, to the segmentation on this 2D image, which is often not an ideal perspective, but then we can also use this to project it onto the 3D model. And in this way, figure out what the plant parts are. So in this case, we have implemented a deep learning network, so with a convolutional neural network. And then on purpose, I've added the paper down here. But the essence is also, and that's why I brought it up also for my colleagues actually, that you need data to train your model. So if we, in this case, this paper describes a very simple plant. So it's just 2 leaves on a stick. But if we are going to talk about the tomato plant, the bigger plants, and it's way more impossible, or way more difficult to figure out what the individual plant parts are. And especially if you would like to go for deep learning, then you have a big challenge in generating sufficient data. And if you are able to generate data, then you still need to label all that data. So the whole network needs to know to train which plant parts are which. So this manual labeling will then become a big challenge. And that's why I brought another project actually to this seminar. In this case, we're talking about a robot that with the aim to harvest peppers. And there's a camera in its hand that is analyzing with RGB and depth information where this pepper is to harvest the pepper. So this is the aim of the robot, fully automated harvesting of peppers in a greenhouse. But you can also see that the environment is completely chaotic. There are a lot of leaves, stems, wires, objects that need to be identified with by the robot to figure out where is this particular pepper that is not only there, but also where is it located? Is it ripe enough to be harvested? And actually a colleague of mine, Ruud Bart, came up with a very smart idea. I think that's very worth sharing also with this audience. So what he actually invented is he actually tackled this data challenge, because he generated a virtual plant model. So what we are looking at here is not a real plant actually, but this is a generated artificial plant with artificial plant parts, leaves, and peppers. And because he was generating it in a virtual domain, he knew already which were the plants, leaves, stems, and peppers. So he could generate training data for an AI approach. So he completely jumped over the problem of labeling data. I'll continue the video. But by generating this data, he could train the robot. So this is one of the images that he was able to generate, which were actually quite identical to the real stuff. So we did train the robot a bit, so we had to retrain the neural network, but actually it was pre-trained on such a level that it could be launched actually quite quickly and easily inside this greenhouse. And then in the end, the individual peppers were found, and color was the indication if it was ready to be harvested, and then the robot went for it. So also in this digital twin approach, we will have to deal with generating data. And we also hope that we can generate data with the virtual plant model approach that Pieter showed in the earlier slides. And one other aspect actually of challenges of data. So you can imagine that if you have hundreds of plants growing, you would like to image them every day, you will end up with not only gigabytes of data, more terabytes or more amounts of data. So we are now implementing that. These are the 6 modules of NPEC. So for instance, this one is the greenhouse with all the different imaging systems. The data is collected on individual computers that are connected to the cameras, but then all the data is automatically transferred to a cloud. There, all data is combined. So we got multiple sensors brought together in one, you could say, big file storage system. And there also, for instance, the algorithms extract information. So we're going to talk about massive file storage for the images, but also information management system to give the end user, so that's actually us, the researchers, access to all the different components that are collected. So also the climate data from the greenhouse or the weather data that could be of relevance for understanding and giving input to the digital twin. So also I could share more information on this, but I think because of time I will move on. But one thing maybe it's interesting for you to know that we have installed and are testing also iRods to automatically do the file transfer, the checksum, and also to simply manage all files. Automated deletion after a certain amount of time. If it's not used anymore, it is able to shift that to cold storage, which is cheaper than keep putting everything in a live cloud. I think, Pieter, it's time to go back to you to continue talking on this diagram.
Yeah, thank you, Rick. Very interesting. Some things were also new to me because there's a lot of technique involved. As a biologist, I really want to know how the crop performs. And growers also want to know, are things going right? Is the plant healthy? So we want to get that information. And in this design of the data flow, you have to realize that at least every day this cycle is done so that we know each day what is the status of the plant. And we have models that predict plant growth. We will parameterize the models on the run by these images that have been gathered, and we know quite a bit. Also, the French scientists, for instance, worked on 3D models of plants where they had relationships of leaf growth, leaf extension, fruit formation, things like that. We think we can derive all those things from these data, and so we will end up, when we do this trial, with a good model that predicts biomass and fruit production under these circumstances. And of course, this year we will get a lot of data, and we will try to make something out of it. In the next slide, you will see what is actually becoming of this digital twin. This is a representation in 3D of the greenhouse compartment, compartment 2, where you see the real structure is put in. The manufacturer had a sort of SolidWorks 3D CAD/CAM image, and we incorporated that in our plant modeling software. We put the plants inside too on the conveyor belts. The next slide, you see that we can also simulate the scattering of the light through the glass cover. That affects, of course, the light's quality, the spectrum, and the direction of the light. And well, this will actually be updated every day if we run the trial. In the next slide, you will see that we get also close-ups of the plants and the pots. In this case, it is made of previously scanned plants that were used for testing the Maxi Marvin and the Robin. The real trial will start in a few weeks, and then we will get the real actual plants. So in the coming months, we will do this experiment. We will use about 3 genotypes that have different shapes of leaves and different sizes of tomato trusses. So we will look if the scanning devices and all the other devices, sensors, will notice a difference between the genotypes. And this will be a very exciting moment.
Yeah.
I don't know what is the next slide. I think
no,
this,
it's fun. I think it's fun for a second to go back to this slide and mention a few. I would like to show a few of the aspects. I'm not sure. Is it? Can you see the individual words?
Yeah, roughly.
Yes.
Okay. Yeah. So in the end, so NPEC. I mentioned NPEC here. So we could say the imaging systems need to deliver, for instance, information on stem thickness, leaf area, internode length, fruit size, ripeness. So that's the phenotyping information. But on the other hand, the climate monitoring tools need to offer information on temperature, humidity, CO2, ventilation, screen lighting, illumination, but also aspects of the outside world, so the temperature outside or the wind speed outside. But also more static information is used also based on this greenhouse with the parameters over here, which is then all fed to Growim, the modeling tool. So everything is actually connected, as Pieter also explained. But within this NPEC Greenhouse, then we are able to give this on individual plant level, while the grower would probably steer his crop more on for instance, row level or on compartment level. So this enables us to give a much more accurate prediction also what's happening based on varying weather conditions, but also varying experimental design aspects.
So indeed, we have about 600 plants in that compartment, and every plant will be measured and scanned at least twice a week. So we know every detail of its growth, and there we will probably find that there will be side effects, border effects of the windows, of the walls, all kind of things that relate to heterogeneity of the climate. A lot of things that are normally ignored in a big greenhouse, but are probably very relevant.
Yep.
Yeah.
Okay, so that's this is the last slide. If you have questions, ideas, or suggestions, this is your moment, I would say.
Okay, thank you very much.
Thank you.
Thank you.
Wonderful audience.
So, are there any questions? Maybe I can start with some. First, thank you very much. I think it's really exciting domain, like the interaction of modeling and robotics and computer vision. Like you could almost see the greenhouse as a robotic system where you use control based on model to drive growing of plants. It's really fascinating, and it opens many ideas as you showed, like the interaction between models and deep learning, how you can use models to train deep learning models. Sorry, is very exciting area. And I was interested in also the 3D analysis you showed. And I wonder, do you have any ideas on how to make correspondence between two scans? You've got your plant at time t, you've got your plant 1 week later, and I guess you want to measure the growth of leaves and etc. Yeah. So did you already tackle this problem of making the correspondence between the two plants?
No, we took in a lot of requirements to make this happen. So the plants are in a carrier, and the carrier is often always exactly positioned in the same way as 1 week earlier.
Yeah.
So in principle, if we have a sufficient short amount of time in between of the scans, then the scans would be quite similar. So the leaves will be extended only a bit. But actually, to do this in practice requires a fully automated phenotyping pipeline, which we have not done yet. So there is a postdoc working in this project also specifically on this 3D data analysis approach. Yeah, so the example that I've showed were the robot systems where the 3D reconstruction was made. There was the focus on individual plants and processing, so not keeping track of individual leaves over time. So that's not done yet, but we took into account that it's potentially possible.
Okay, thank you.
Yeah.
Pieter, you have a question?
Yeah, I'll turn on my camera for a sec also so you can see me. Hello, thank you very much for the presentation. Very interesting. and it's also extremely relevant to us. I don't know how much David has talked about the work that we're currently doing here at Sony CSL.
Very short.
So one of the things we do is indeed the 3D imaging of Arabidopsis thaliana to look at the stem shoot, to measure the angles between the fruits and the internode lengths between the fruits. And of course, one of our references is your work. So it's really interesting. It's really nice to be able to exchange with you today.
Oh, cool. Yeah.
And so I have a couple of questions. I mean, I hope I wrote them down in no particular order. One of the first things that came up, so you used a fixed set of cameras, so that are on a fixed infrastructure. We're working with the mobile camera. Did you have any experience, like the advantages of using fixed cameras compared to, say, a mobile camera?
Yeah. Our approach is very rigid. So we have a very accurate calibration procedure. So each individual voxel is either a plant or part of the background. So there's no uncertainty there. So if the segmentation of the 2D image is fine, background plant, then the model is quite good. So we know exactly that we can do this because we know the exact position of the camera. So we have been teaming up with also the people in the University of Nottingham, Tony Pritmore, and we actually also, he has a camera mounted on a robotic arm, and he has also a completely different 3D reconstruction approach. Because if we would do our approach, you could say the Marvin calibration approach, and we would need to have a robot system that is really accurate. So it needs to be able to position itself exactly on the same spot down to sub-millimeter level. Otherwise, our approach does not really work. So we've been testing this, and I think the cameras are affordable, and we decided not to go for this robot arm approach, also because of throughput. So in this case, we can the scan really takes only a second, actually a few milliseconds. So high throughput is in this case more important than saving some money on 14 cameras, you could say.
Yeah. Well, indeed, we have the problem of correctly positioning the camera, and we use software, actually, structure from motion to estimate the correct positions of the camera, but it requires quite a bit of computation time.
Yeah, so there's a colleague of mine that's making beautiful pictures, high resolution pictures, multiple angles, really a lot of pictures, and he also generates beautiful point clouds, but it takes long to generate. So physically it takes long because you need to move the plant, you need to wait and move the robot arm, but also the calculation time itself is also quite long. But if that's fine, then for your application it works. But
well, to add to that, Rick and Peter, when we go with this system to a real greenhouse, you have to realize that you cannot make a system of a plant towards the sensor, because those are fixed, those plants, and they are very tall. So then we need to have a camera going to the plant, and we face the same challenges as Peter.
Yeah, definitely. Then we go down to the sweeper robot approach. Yeah. But then the accuracy of your models will be quite poor, I'm afraid, because it's such a big mess of leaves and stems all over. So then it depends what do you really want to measure. We don't actually know yet what we really want to measure. So we want to generate a beautiful 3D model and then figure out what's relevant.
Yeah, it's not totally correct, because there's also a question of Aldo, I see, on the registration of the plant growth and what to do with that. Well, there are some plant modelers in the group, and we realized that we first want to know more on the response of the organ growth to temperature. That is basically the first you do when you parameterize a plant model. So we look at the extension of leaf 3 or something, given the climate in our NPEC greenhouse, and then we make this into the model and calibrate it. Second thing is leaf area and weight of the leaves and stem. We can try to extract that from the images, but volume is, I think, not so very accurate.
No.
I mean, it would be very nice. Then you can use the density to predict the weight of the plant. But we will do some destructive measurements in between by the postdoc. And so we will, in a couple of weeks' time, be able to parameterize two very important aspects of the plant model: the temperature response and the response to light and photosynthesis. So there is really a thought about that. But for now, we will have the experiment and loads of data will come in. I think after a few months, we could come back to you and say whether we successfully calibrated the model.
Yeah, and we have different backgrounds, so this is also a great arena to validate if we agree.
Yeah.
Yeah, we are learning too. Yeah.
Yeah.
You are muted. Yeah.
You're muted.
There's another question. So this relation between data gathering and the plant modeling. So what would you think would be, I guess, midterm perspectives on this, in the sense that how far? First question would be like the GrowIMP. Is it a biological model of the plants, or more like a like phyllotaxical model?
Yeah, that's a good question. It is a biological model building from the basis upwards. So really growth of organs, carbohydrates, translated into sizes and shape. So not a describing model where the pathological processes are a black box. This is really an open box. And I know there are also models that are more statistical and descriptive. You could also parameterize those, but since we want to give decision support to a grower on its climate, temperature, humidity, radiation, it should have a physiological basis. So that's inside of the GrowIMP. It's FSPM in its fullest form.
Okay.
I don't know if you had Arabidopsis models. I know some Arabidopsis models on their 3D shape, but they are also more or less descriptive, I think.
Well, I can answer that quickly. We're working with a group from INRIA. It used to be the virtual plant groups. They renamed. It's the Mosaic group. It's headed by Christophe Gaudin.
Yeah.
And so they have a biological model of Arabidopsis based on a rule rewriting system. And so
it's the same type of software. And I know Christophe already from the FSPM community. Yeah. Interesting. Yeah.
And so, the next question actually also would be like, how would you see so once you have a model, you can start using it for prediction. So how much did the plant grow? How much you expect the plant to grow? And then data coming in, and then trying to match the two, possibly using the comparison to improve parameters of your model, even of an individual plant. Do you see that something that is feasible in the relatively short term? Is that something that you plan to do?
Yep.
Rick, it's also a question to me, I think.
Yeah, I think so.
Maybe you can add. In our business unit, Greenhouse Horticulture, we use already for 15 years a process-based model. It is parameterized for a couple of important greenhouse crops like tomato, sweet pepper. And what we lack there is its structure, its 3D structure, what we actually need to determine its light interception and photosynthesis. But it's already pretty good validated. And that model is already used to support growers on how to do their climate. And for instance, to say that the lamps can be put off, because there is enough radiation, or yeah, things like that. So we think that we can improve that existing model with bits and pieces that we develop now. And so that is already a little bit on track. Right? But I think this system that we showed now is too expensive to be carried out, of course, by a grower. But they can, I think, use some parts of it, isn't it, Rick?
Yeah, I think also what I'd like to mention, so we have been working on fully autonomous greenhouses, where the grower is simply overtaken by an AI. We have organized really a competition. Have you heard of it?
No.
Okay, so it's really like a competition where we say you will get a greenhouse compartment, for instance, with cucumber plants in. We've done that now for the second time. The third time is coming up. It's a big event, actually. A lot of these bigger companies also join. So it's also companies from China, like Tencent is participating, but also Microsoft and others that try to beat the human grower. So there are 6 teams, 5 of them are AI, and the 6th is a human expert. And then they are trying to to optimize the yield while also taking into account the energy consumption. And for instance, there is a hackathon, a pre-selection, to figure out which teams are actually quite good to go for the real experiment in a real compartment in our greenhouse. And this hackathon pre-selection is done actually that they are working with our greenhouse model. So the greenhouse model is used to figure out if their AI is good enough to run a greenhouse at least in a decent way. If you completely mess up in that model test hackathon, then you're out. If you score high, then you're going for the next round. But I think this model is, and also this AI approach is very much based on controlling the temperature, humidity levels, some basic, you can press basic parameters on the long run. So over the course of a whole growth season. What this digital twin is now focusing at is really to dive into the plant and really understand what actually the plant is receiving on the amount of light on the leaves and the leaf area and leaf expansion. That's not part of this autonomous greenhouse concept yet. So that's definitely a super big next level jump that we are now aiming for. It's not for sale yet for a grower. It's really fundamental, you could say, fun. But in the end, it will be handing out a more detailed analysis of the crop to a grower or even a whole breeder.
And sorry, how is this competition called?
I'll put a link in the chat. I think
Okay,
that works.
And if
here
I
it
may,
will be on lettuce.
okay.
This year it will be on lettuce. Last year it was on cherry tomato. First year it was on cucumber.
Okay. If I may, just one last question, and then I'll just I'll leave the floor to others. I'm a bit intrigued also by the Ecotron and whether there's any imaging devices that are used to study, say, groups of plants. It's a question that we sometimes have in the team because plant association is something that's being discussed for, say, sustainable farming, but it's really hard to study. And I was wondering whether any of the imaging technologies was also used for, say, plant associations.
Ooh.
I don't know.
Now, can you maybe repeat the question? I'm not sure if I understand it right.
So it's it's more related, perhaps, I guess, to the Ecotron that you also mentioned in one of the slides, where you set up, say, associations of plants, and so if you want to study the effect of putting plants, different varieties together, and whether any imaging technology is already used to follow up, say, the volume or the height or the biomass of the plants in these more complex situations.
Yeah, you mean like plant to plant interaction? If they share the same soil, the root system competes, or maybe the one overshadows the other one. So it is actually backed up. I saw it a bit in GrowWimp, where you can also simply put different plants next to each other. I think Pieter, was maybe in one of your slides that you saw different types of plants next to each other.
Oh, absolutely. One of our colleagues in the Digital Twin project works on inter-row competition or interspecies competition. And you can very nicely use the 3D models to look for light competition. But the question relates to the images in real time that were performed. I don't know. I think they made photos and they did light measurements inside of the canopy. I'm not aware of any automatic camera system.
Not as such. I think the Ecotrons, they are not installed yet. They will look at, they can look at these kinds of plants setups with multiple species within one tiny box, which is in a very controlled condition. Their focus, actually, main research is on plant-microbe interaction. But on the other hand, we are also running big trials, but it's on the fields. And we focus on intercropping. We have long single paths of potato next to weeds and next to whatever, and then we figure out if there is some positive effect on the fact that different crops are next to each other while they are still harvestable in an automated way. But then what GPS-based harvesting of single strips. So it is being done, but I think not exactly like you describe.
Okay, thank you.
Yep. David, now you are muted.
Yes, sorry. Is there any other question?
And I also want to mention that Bolai is actually in the room. That's our postdoc working on the 3D models. So. There he is. Yeah, cool.
Maybe I have one last question, if I may. You mentioned some light treatment when you do fluorescence measurement. Could you precise a bit what kind of input you send to a plant?
Yeah, so I'm not an expert on this topic, but I've been like watching what they have been doing. It actually, for instance, about the study of where they found actually in Arabidopsis a mechanism, and also you could say how quickly a specific variety was able to switch on the photosynthetic mechanism again. So if, for instance, there is shading by clouds, and the plant lowers its photosynthetic mechanism, and as soon as the sun comes back, it switches it on again. And there's a paper but that's from the genetics department, and they figured out with this phenotyping tool, another one also within NPEC, that there were differences in this mechanism between plants. But then they were giving specific light treatments, so switching off light and on the light, or a bit longer or a bit lower, or different quality lights. And they looked at the response of these. Could you imagine a whole table of a whole batch of varieties, maybe like of varieties with genotypes, 1,400 plants, all individually measured by these camera systems. And by looking at the phenotype, they could make a link with the genes, and they could figure out that they could optimize actually the recovery speed, which was directly linked to specific genes. So they could actually breed plants that could grow quicker and were more responsive to light. Right, like optimizing varieties for light, like light efficiency.
Okay.
Yeah.
Yeah.
Is it correct, Pieter? I think you know more about it than me. But,
no, I don't know much more.
Correct, maybe.
I think the question was also what kind of wavelengths do you use, but you probably also don't know.
You can make your own light recipes.
Yeah.
So it's I'm you could say the NPEC project manager, enabling people to make use of the tools, and the people that are making use of this particular imaging system are really the super experts that I'm not able to keep track of what they are capable of.
Okay.
We have a range of lighting systems in there to give treatments to the plants. Well, that's on the web. I can give you a link actually if you find more information on it.
Yeah, if you have a pointer to that paper, we'd be interested.
Okay. Yeah. So one of the camera systems in NPEC Greenhouse, the chlorophyll fluorescence imaging system, is by the company Innovation. But the Robin PSI is also on the web at the NPEC website. And I'm gonna search for this paper by Mark Ice. I'm gonna put it in the chat actually.
Okay, thank you very much. Maybe for the paper, because we are closing the seminar, you can send it to me and I will transmit to Pieter.
Okay, I have it already. It's down there. Yeah.
So thank you very much again. It was really nice seminar, and I hope to see you all next week, where Bharat Raghavan will talk about computational agroecology. So to see you there.
Yeah.
And have a nice evening.
Yeah.
For Europeans.
Yeah. Thank you
you
for inviting us. Yeah.
Thank you for your attention.
Thank you very much.